So going back to a very quick overview, as we all have been involved in various aspects of virtual bioequivalence or assessment of different scenarios, we are coming into different scenarios, different cases that we would like to assess various clinical scenarios. And then the question is usually, can we simulate or predict various clinical scenarios of the same formulations or different formulation and compare relative exposure under either a bioequivalence statistical framework or an arbitrary statistical criteria? And the reason is that not always we are looking at the bioequivalence. This is a publication where the different scenarios they were compared. For example, in one case, 300 milliliter of water was given. Another case, 330 milliliter of Coke was given with the same formulation. Another one, water with proton pump inhibitor, or Coca-Cola with proton pump inhibitor. And not necessarily always it is possible to run all of these scenarios, and it is important to see whether formulations are working the same way or the same formulation or different formulations are working the same way, and whether they are bioequivalence or not. So, and as you can see, it is not necessarily about specific formulation, but it can be different scenarios. So what we can do, we can have a framework or a platform for assessing virtual bioequivalence. There are different elements that are necessary for doing that. Obviously, finding the most sensitive or the most important parameters through a global sensitivity analysis or local sensitivity analysis, having a good mechanistic absorption modeling, and identifying and incorporating between and within subject variabilities. These are some of the things that Ioannis is going to explain in his presentation. Incorporating critical quality attribute as well as having a framework to assess those is very important. As you know, we have been developing the absorption model for many years now, and we started to introduce the SIVA as a way of analyzing in vitro dissolution and solubility parameter or experiment, and get some parameter and incorporate them within the ADAM model. And another thing that we have to do in the context of a virtual bioequivalence, we have to incorporate critical quality attributes and inter-occasion variabilities, within subject variabilities, between subject variabilities, because we may give the dose or formulation at different occasions. And then we will run through and incorporate those factors and assess the virtual bioequivalence. And this one can be expanded. It can be expanded, and it can be an automated way of finding the safe space, which can be very important aspect of this expansion. And we are moving in that direction. So this is the general direction that we are taking. And in version 20 of the system simulator, we will develop a virtual bioequivalence add-on module that it allows you to cover various different scenarios. And many of the commonly used studies, they have been incorporated. So crossover, like 2 treatment, 2 period, and 2 sequences, which is the typical bioequivalence studies, it is incorporated. And there are others, like 2 treatment, 2 period, and 4 sequences, or crossover partial replicate, full replicate, different sequences, parallel as well as customized ones. They can be incorporated. And then you have the ability to add within subject variability or inter-occasion variability parameters to some of the physiological parameters. And then you can run and the data seamlessly are transferred to Phoenix for bioanalysis. And the reason we were explaining this one, because this leads to the Ioannis presentation, and how this can help with those type of assessment and make it some kind of automated, so reduce lots of manual calculations and allow incorporation of the various elements that I mentioned. At the moment, there are a range of parameters from oral as well as dermal route administrations. they can be incorporated in the subject variability. I'm not going to the whole list. In terms of the work, it's similar to the way that we do sensitivity analysis and parameter estimation. So different pH, gastric emptying, transit time, and surface skin pH, they are incorporated within the module. So it make it relatively easy to be able to incorporate those. And then now we are working on adding other formulation-related factors that may change from occasion to occasion. Okay, that was a quick introduction to virtual bioequivalence. I'm going to we are going to move to the presentation. I'm very glad that Ioannis have accepted the invitation to present. Very interesting case study. And Ioannis just finished his PhD in Jennifer Dressman group, and he's a senior scientist in Novartis. And he has done a number of different publications, and as you will see, he's going to take us through the different cases, as well as he's presenting a very good introduction to the approach and various complexities that we are going to encounter when we are doing virtual bioequivalence, different approaches, and any other consideration that we have to have. I pass it to Ioannis, and hopefully you will be able to ask any question. If you have any question, please feel free to post them on the question, and then as we go through the presentation, you will be able to hopefully hear the answers to those.
Thanks, Masoud. Thanks again for the kind introduction, and I'm very excited to see all the advancements in the VB module in the version 20 in SimSip. So, okay, let's get started. So today I'm going to be talking about, as you mentioned, PBPK modeling and simulation approaches to assess risks associated with bioequivalence in drug development. And just as a disclaimer, that the presented word is not related to any Novartis project or confidential information, and has been already published in the literature. So coming to the agenda, we will going to start with a short introduction to virtual bioequivalence, as you already warmed us up, Masoud. So I will keep that very brief, and then we will see how in vitro and vivo extrapolation in PBPK modeling, especially for biopharmaceutics, can help to establish the link between in vitro, in silico, and in vivo, and ultimately translate differences, for example, in formulation into the pharmacokinetics. We will spend some time on how we can incorporate inter-occasion variability into the PBPK models with some different approaches, and then the main part of the presentation will be dedicated on a paper that we recently published about quantitative approach in vitro bioequivalence based on a risk assessment. And the model drug there was Ibuprofen. And of course, we will close up with some current challenges, limitations, and future directions. So very briefly, we know that virtual bioequivalence is attracting more and more interest, both by regulatory, academic, and industrial partners. And of course, this is because its applicability in a wide range of applications within drug development. Either this concern like new drug candidates or even in generic development. And of course, some of the applications are listed here. For example, when we want to switch between different formulations, for example, from an early phase formulation to a final market image, or for scale-up and post-approval changes to support biowaiver strategies, and of course to explore a possible extrapolation of bioequivalence results to specific populations, if that's pediatrics or patient populations, for example, in oncology, and uh, as you already mentioned, with quite some success in other routes of administration recently, for example, the intradermal administration, or also extrapolation to different prandial conditions or other scenarios where we have, for example, drug-drug interactions with acid-reducing agents. And of course, we have the recently published draft guidance by the US FDA on PBPK analysis for biopharmaceutics applications. And there we saw that virtual bioequivalence was referenced quite some times. So some points that were highlighted also during our IQ commenting group discussions were that, for example, sample size and justified values for between subject and within subject variability were deemed to be crucial for PBPK bioequivalence assessments. And another interesting point was that a non-B batch and or even IV data, it seemed to be that they were highly recommended by this draft guidance, which brought us to a lot of discussions. But yeah, I think that everyone is aware of this current guidance. And if not, I highly encourage you to go and have a look. And in another recent report of 2021, we saw the number of applications of PBPK with regards to biopharmaceutics in NDA submissions, having an increasing trend between 2008 and 2015. In general, the agencies have been encouraging in submission of such PBPK applications, especially for clinically relevant drug product specifications, and of course other applications, including, for example, food effect, using virtual bioequivalence. But as you see, after 2015, let me add my pointer. After 2015, we saw a gradual decreasing trend. This was attributed either that sponsors were very early engaging with the agency to consult, but potentially another reason is the admittedly relatively low acceptance rate in such kind of application, especially when we are comparing, for example, in other fields like the drug-drug interaction. And of course, some of the points that we might highlight here is even there are different initiatives going towards the directions, best practices, and on how the dissolution input is incorporated into the PBPK models have not yet been established, and at the same time having a non-B batch and human IV data to increase the confidence in the predictions, it's not always the case. So moving now to the concept of in vitro and vivo extrapolation, this time in biopharmaceutics. We see nicely here shown from this publication by the Simpson colleagues how stepwise sequential estimation and confirmation of the biopharmaceutic parameters can serve as a more mechanistic input to simulate the in vivo drug absorption, starting always by the aqueous solubility and then moving to different parameters related to the micelle-mediated solubilization, and of course then having different experiments from various biopharmaceutics tools, either those having to do with single phase or double phase dissolution, or transfer experiments, for example. Such an approach, for example, using the SIVA toolkit, an in vitro data analysis toolkit, allow us for mechanistic incorporation of the estimated parameters into the main simulator using the ADAM model to simulate the absorption. And of course, this has the clear advantage of being able to incorporate physiological between subject variability on those parameters, which in other cases, such as if those data were added in a tabulated manner, it wouldn't be possible. A slight caveat perhaps here is that when we are having formulations, especially enabling formulations or complex excipient effects, these are not always very easy to be translated, and they might require a very rigorous approach in terms of biopharmaceutical experiments with proper design and modeling activities. Coming now to the hierarchical levels of PK variability, which are inherent in every study, and that holds true, of course, also for bioequivalence studies. We all know that the GI tract is a highly heterogeneous and variable environment, and in this context, absorption-related parameters can be subject to either variability between different subjects, as you see here. For the same occasion, these two different subjects might lead to completely different or very different, let's say, PK profiles, but also at the same time, we might have variability within the same subject, but between two different occasions. And in general, we would advocate that the between subject variability is reasonably well captured by the current PBPK platforms. However, we're coming in a more challenging situations in terms of the within subject variability, which can be confounded either in the drug substance PK or the drug product performance, or it can be unexplained, for example, in the case of residual variability. So, and the next question that we have to answer is how we are going to incorporate this interoccasion variability into the virtual bioequivalence trials, and whether this kind of variability is it important for the PBPK modeling approach. And I hope with a case example that I will show you later, this will be clearer. So, assuming that the residual variability, as we mentioned before, for example, due to bioanalytical error, is negligible, then we would have two different approaches for the incorporation of interrogation variability into virtual bioequivalence trials. The first approach would be to implement this type of variability into the system parameters of the PK model, as previously mentioned by Masoud, and then propagate that one throughout the simulation. And another approach would be to have prior knowledge about intra-subject variability and then add it to the PK metrics post hoc. So, with regards to the first approach, of course this is considered more mechanistic, but on the other hand there are very limited data available on physiological parameters, especially in the GI and their intra-subject variability. And this is often due to particularly to the design of this kind of GI aspiration studies, which they have usually quite limited sample size and also not that many occasions to get insight into this kind of variability, even though there are some reports in the literature. With the second approach that we were discussing, so in theory it should have been a simpler approach and relying on prior knowledge, for example, from previous bioequivalence studies, that could be straightforward. However, it would require an additional step of adding the variability post hoc. And of course, another caveat here would be that we might have less understanding of the sources of this variability and the decomposition of the different elements. So here you see a typical PBPK modeling and simulation workflow for vitro bioequivalence from a paper that we recently published. And here you see the two different approaches that I mentioned before. Of course, the skeleton or the core of the PBPK model development and validation doesn't change really. So we always have the incorporation of ADME data and of course system parameters to those the variability can be added a priori, and then model development, refinement, based, for example, on clinical drug-drug interaction studies or formulations with different release rates and different PK profiles, model validation, and then performance of the bioequivalence trials and assessment and statistical analysis of the results. So coming to the case example now that I mentioned before, that was a collaborative effort led by the Center for Pharmacometrics and System Pharmacology in the University of Florida, where Rodrigo was the lead scientist, together with Sarah from the University of Florida as well. And then we joined efforts with other folks, Bart, back then who was in the University of Leuven, and now in Pfizer, Amitava from J&J, and Chang from the University of Sao Paulo in Brazil. And the purpose of this modeling exercise was to develop a risk-based approach or framework for virtual bioequivalence assessment, and we have some nice data for Ibuprofen, so that I'm going to show you shortly. So I think Ibuprofen doesn't really need any introduction, but just for like very short background information, we have a typical BCS class 2 weak acid with complete and dissolution limited absorption. The compound is of low clearance, low volume of distribution, and very high protein binding, typical behavior of a non-steroidal inflammatory drug. And of course, we have very high permeability and almost complete bioavailability. So what triggered us for this kind of exercise was performed was a publication back in 1996, where in a meta-analysis it was found that 14 out of 25 powered bioequivalence studies with immediate release formulations of ibuprofen failed to meet the bioequivalence criteria, especially for Cmax, but not for AUC. And based on that one, we decided to perform this PBPK modeling exercise to explore the role of interlocution variability in virtual bioequivalence, and also provide a tool that we think that it can be helpful for teams to take go no-go decisions and also inform prospective bioequivalence studies. So as mentioned before, we started with in vitro data, in vitro biorelevant dissolution data that were performed back then during the PhD of Rodrigo. And then we we performed some um, analysis of the in vitro data using the SIVA toolkit. Then the estimated parameters were transferred into the other model in the SimSip simulator, and then with estimated particle size distributions from the different formulation with the different dissolution, we simulated the absorption and the PK, and after that we perform bioequivalence trials. In the first step you see without the addition of interrogation variability, and in the second step interrogation variability was added post hoc. So moving to the details, we will start with the in vivo bioequivalence data. So we are talking about two studies of two period, two treatment crossover design, where the enrolled population were fasted healthy adults. And actually we had a reference listed drug product, a generic product which was approved by the Brazilian Health Authority, and a non-bioequivalent biobatch. And here you can see the geomean ratios with their confidence interval. So for the study 1 and the bioequivalent product, both Cmax and AUC met the bioequivalence criteria, whereas for the non-bioequivalent batch, we saw that both Cmax and AUC fell outside the bioequivalence limit. And in this column you see also the estimated within subject variability that we had from those studies. Interestingly, there is slightly lower intra-subject variability in the study with the non-B batch. But we will come back to that. And then we had the in vitro dissolution data. Again, these data are published by Rodrigo and Jenny in the Journal of European Journal of Pharmacokinetics and Biopharmaceutics. And the in vitro condition used here to provide the enough discriminatory power was with a USB2 apparatus at 50 RPM using an experimental version of FASIV named FASIV version 3 and reduced concentration of phosphates to actually better match the buffer capacity that is observed in human aspirates. And in addition to this experiment design, also as it was observed that the surface pH of Ibuprofen, it's critical for the microenvironment and dissolution. The bulk pH was kept constant at 6.7, which is the bulk pH of FASI version 3 by titration when that was necessary. And here you can see that using these dissolution conditions, the three different formulations were nicely discriminated using this dissolution method. So coming now to the PBPK model, and I'm mentioning here PBPK model refinement because the initial model was validated and published already by previous work by Rodrigo and Jenny. And what we did in this case is that we updated the model by estimating product-specific particle size distribution based on the in vitro dissolution data that I just showed you, and then using the estimated particle size distributions, we transferred those to the other model, and additionally we used the particle population balance model, which in principle assumes that the solid drug particles shrinks when they are dissolving. And of course, I will just show you in the next slide the confirmation of the model ability to describe the absorption of the different formulation and the performance. So you see in the table on the right, overall the predicted results were in a good agreement with the observed, within 1.25-fold, and this gave us enough confidence to move forward and use those inputs to further simulate and perform these virtual bioequivalence trials. And also, as you can see, we observed also a nice formulation writing by the PBPK model predicting the differences between, smaller differences between um, reference and bioequivalent formulation versus reference and non-bioequivalent. So in this slide, you see the results of the bioequivalence trials. So the trial design that we used were a standard 10 trial design where the sample size was equal to the sample size of the actual studies, so 23 for study 1 and 35 for study 2. And also for the simulation setup, because for these simulations we used the SimC version 19, in which the virtual bioequivalence module was not available, so we used the fixed sheet to somehow replicate actually the 2x2 crossover design. And in this case, as I mentioned before, in this first step, we didn't add any interrogation variability. So any differences that you see here, for example, in the point estimates, and here I'm showing the point estimates for Cmax at least, is attributed to the differences of the formulation because we have exactly the same disposition and elimination parameters for the subjects. And then what is interesting to see was that between the bioequivalent product and the reference, solely based on the dissolution, we had an S2 of 53, showing similar dissolution, and this resulted in this plot here in a Cmax ratio ranging between 104 and 106. Whereas, even though we had the similar dissolution between the reference and the non-bioequivalent formulation, the Cmax ratios predicted based on the PBPK were between 110 and 115, which that would mean that would lead us to a biased result of bioequivalence where we would know a priori that this was not the case in vivo. And this brings us already to the second step of adding the interrogation variability into our results post hoc, and to do that we performed some bootstrap analysis in the interrogation variability that we had from the in vivo studies, and we obtained the bootstrap confidence interval, 95% confidence interval, and as you see here, the confidence interval between the bioequivalent and non-bioequivalent formulations were overlapping based on the bootstrap, and this led us to believe, or at least to assume that so the main element here in the net interrogation variability was not due to the absorption, but mainly due to disposition parameters. And for further assessment of the virtual bioequivalent results, as a worst case scenario, we used the upper limit of the bootstrap confidence interval, which was around 20%, as you see here, for the Cmax at least. So addition then of the interrogation variability to the PBPK estimates, and then recalculation of the confidence interval based on those point estimates that we had from the PBPK results, nicely now discriminated between the bioequivalent formulation and the non-bioequivalent formulation. So you see that the range here in the confidence interval, it's broader than the 10406 that we see here, but still within the bioequivalence limits, and nicely now we get that indeed the non-bioequivalent formulation was proven non-bioequivalent, at least in terms of Cmax. Also, if you remember the upper limit for the in vivo study that was that failed to meet bioequivalence for the Cmax was 135, and here we see nicely the PBPK model coming quite close to that value and picking up the right result. So coming to the last part of this case example, what we wanted to do more is, given 20% of interrogation variability as a worst case scenario um, for Cmax, we wanted to calculate what would be the adequate sample size that would give us enough power of 80% for those bioequivalent studies, given the point estimates that we have from the PBPK prediction. And as you see, for the bioequivalent formulation here, a sample size of even 24 or 36 would be sufficient to reach power of 80%. Whereas in the case of the non-bioequivalent formulation, what we see here, at the best case scenario that was predicted for the point estimate for Cmax from the PBPK, which was 1.1, we would need at least 36 subjects to power properly this bioequivalence trial, and in a worst case scenario, we would need up to 120 subjects if the true point estimate for the Cmax ratio would have been 115. And also here you can see, for example, with 36 subjects, we would reach only up to 0.2 power, so only 20%, even with 36 subjects. And such a kind of approach, we really believe that could be useful for example, for clinical teams to inform go and no-go decision making in terms of bioequivalence, and also design prospective bioequivalence studies in the context of performing some risk assessment around different, for example, IOV scenarios or point estimates, and then calculate what would be the required sample size and what would be the risk, for example, to perform a bioequivalence study with a smaller sample size. So coming to the model limitation and outlook, even though the current model was able to reproduce the in vivo bioequivalence results in terms of Cmax, that was not the case for the AUC. That was a little bit of surprise in terms of ibuprofen is typically expected to not to be sensitive to the AUC ratio because of complete absorption and very high permeability even at the colon. But nevertheless, we thought perhaps other mechanisms such as, for example, propagation of interrogation variability and clearance could help perhaps to get also to reproduce that result as well. Another possible limitation going to the same direction could be perhaps that we had a simplified elimination component using IV clearance and enzyme kinetics, for example, with uncertainty could perhaps alternatively give us some more reproducible results in terms of the AUC ratios at least. Of course, further validation of such an approach with different compounds and or B studies would be required, and of course the appropriateness of such a kind of analysis highly depends on properly designed in vitro experiments, on the biopharmaceutics tools, and the analysis of their results, and often can be a case-by-case approach based on the compound biopharmaceutical and formulation properties. Um, some current challenges in virtual bioequivalence. Um, one major is that omission of interrogation variability, which I believe has happened systemically in the past, or even inclusion of the interrogation variability, but with arbitrary values, might lead to biased B predictions, and that can be especially true for highly variable drugs. Um, as mentioned before, little is known for the variability of those parameters in vivo, especially for the GI tract. Um, More complex designs were not really possible, so now with what Masoud showed, that perhaps is not anymore the case. Um, it still remains quite challenging to decompose the different sources of IOV and also um, entirely understand the different complexities due to excipients or due to enabling formulation, as well as their interaction with the GI environment and its variability and how this can be captured by the PBPK models. And I'm referring only to the fasted state because if we move to fed state or even other populations, um, this picture might get even worse. Um, yeah. So what would be some future directions or some opportunities that we have at the moment is, of course, a more in-depth profiling of the GI tract would, of course, be beneficial with properly designed in vivo studies that are able to reveal this kind of physiological variables, the variability, the distribution, and possible intercorrelations. Um, to explore further different sources of interrogation variability, and that's something I think that can really be performed using a PBPK modeling approach, just to get what affects more the PK, even though we might not have full understanding of the in vivo values. Of course, we need more examples and further validation of these approaches. Yeah. So coming to the end of my presentation, if there is a take-home message, would be first that an IVIVE-PBPK modeling approach in biopharmaceutics can help to provide a more mechanistic framework for establishing the link between in vitro, in silico, and in vivo, and translate those results to differences in PK. Of course, interrogation variability, and I hope now that's more clear, it's integral part of bioequivalence, and any omission of it or arbitrary values will likely lead to biased P predictions. And lastly, that virtual bioequivalence trial simulations, such the ones that I presented in this case example with the addition of IOV post hoc, and then some power curve analysis and sample size calculations can really serve as a helpful tool, a quantitative tool to inform teams about decision making and design prospective bioequivalence studies. And at this point, I would like to thank the Center for Pharmacometrics and Systems Pharmacology from the University of Florida and Rodrigo for this great collaboration. Sarah, also from the University of Florida, Bart Amitava from Pfizer and J&J, Chang from the University of Sao Paulo, of course, the European Training Network Pearl, and my PhD supervisor, Jennifer Dressman from the Goethe University. Masood and David for always the lively discussion and their continued support, as well as Heidi and Birgit from the Novartis Modeling and Simulation Leadership, as well as the whole Novartis Modeling and Simulation team, and last but not least, you for your attention, and I'm very happy to open the floor to any questions.
Yeah, thank you very much, Ioannis, for very good presentation, very informative, covering various hot topics, I would say, in the area. Any question from the audience? Either you can just raise your hand so we can unmute you and you can ask your question, or you can type your question in the chat box. I think while we are waiting for the question to come through, I think you highlighted a few very important points. And for example, identifying the interrogation variabilities is one, and you very clearly show that if we are not considering those, or we use arbitrary numbers, then it can give us a false type of a result. But what do you think is, or how can we go about them to find this type of values? What type of study do we need, or do we need more tools, or who can provide those type of interrogation variability?
Yeah, I mean, I think that's a fair point, right? For sure, we are. I believe that we are really depending on more in vivo data coming, especially when we are talking about the GI tract, right? There are like some information published in the literature about different occasions in actual B studies from the University of Greifswald. But I think really we need ideally either more in vivo studies properly designed, because it's quite often the case, at least to my knowledge or to my experience, that aspiration studies, for example, are statistically designed in a way that it doesn't capture like this kind of different occasions and getting insight into variability between the different occasions. So perhaps I think one point would be to understand more the GI profiling, and not only in the fasted state. I mean, that's even more difficult in other prandial states, or either disease or specific population. And also in this case, for example, I really believe that having a tool like the SimSip VB module could be helpful in the context of, for example, you can simulate different levels of interrogation variability in different parameters in a kind of sensitivity analysis, let's say, and try to look which seem to be the more sensitive, and then perhaps go back and design an in vivo study and possibly confirm your assumption. That's maybe what I would think would be a way forward, but for sure it's not something easy to do, I would say.
Yeah, thank you very much. Is there any question or comments from anybody on the, as I said, you can either raise your hand, we can unmute you, or you
Questions
can,
just come through in the chat, Masood. Do you want to read that out?
I think they are to you, Luke.
Oh, sorry. Yeah, sorry. I'll read it. The FDA seems interested in Bayesian population PBPK modeling, integrating ISV and IOV from clinical data in a PBPK structural model. What do you think of that approach?
Hmm. So how do you mean it exactly, like in a structural model to incorporate it?
Hold- Frederick, I've unmuted you if you wanted to explain that.
Yeah, sorry. Yeah. Well, it would be using a PK model like any model in a population PK analysis, except it's a more complex model, and you would probably have to calibrate a reduced set of parameters. But in a way, what you would do is integrate the IOV and the intersubject variability also from your clinical data into the parameter of a PBPK model, rather than having just prior information or having just bottom information in your PBPK model. And instead of adding IOV at the end as an added variability.
I think it makes sense. I'm more thinking to which parameter you are going to assign this IO. I guess this is like the critical point that we are discussing now, that we might not know which source it's really responsible for this IO, and how you have, to which parameter you have to implement it, or parameters to get the reasonable results.
Yeah, the identifiability question seems important. Yes. Okay.
Amit, did you have something to say? You've got your hand raised.
Yes, I just wanted to clarify, maybe Frederick and Ioannis also can comment, but what FDA is asking, I don't know whether there are any FDA colleagues that they can clarify, is actually in line of the typical classical PK, because interoccasion variability that you are talking about, Frederick, they are related to the parameters, as Ioannis was saying, that we are getting from the profile. What we are talking about is the interoccasion variability of the physiology, nothing to do with the drug. So I don't think, honestly, they appreciate perhaps, the differences that we have got in these two. Interoccasion variability of the features of the PK that we are capturing from clinical studies, one thing, but interoccasion variability of the physiological parameters that they are leading to the interoccasion variability we are observing on the PK features is a different thing. I don't know, I'm confusing the issue more, or whether it is clarifying, but I think there is something here that we need as a community to actually work on more, because these are mixed up. Whenever we talk about interoccasion variability, unfortunately, because of the PK community's view on the interoccasion variability, they immediately start thinking of the interoccasion variability of the features of the PK, the observations, rather than the interoccasion variability that we are talking about of the physiological parameters that propagate and cause these observations. So, these two are very different, and I cannot see how actually you can deal with them via the observations, analysis of data, unless actually Margarita is already on the call, and she has been looking into possibility of, Ioannis mentioned, that looking at different combination of physiological interoccasion variability and see how many of these they are compatible with the observations. This is not absolute solution, but then you can find it saying, okay, this kind of interoccasion variability, if you assign to stomach emptying, is never compatible with what we are observing. Or this level of interoccasion variability in pH is not compatible with 50% of the drugs that we are observing. So you are basically exclusion criteria for what is possible or potentially is there and what is not there.
I fully agree with this comment. I believe perhaps what was mentioned, it's in a classical PBPK approach that we might have, let's say, estimated the interoccasion variability, and then I would assume that perhaps they mean that you use that again as information prior to simulate adding this kind of level of interoccasion variability, but of course that's something different from what we're discussing here, how the physiological variability translates to what you see in vivo. So yeah, I fully agree with
The key, as you know better than me, would be that in that case, you are assuming that is going to be constant between any formulation that you are putting. While we know when the formulation changes, their susceptibility to the physiological interoccasion variability are going to be different. Therefore, you may have, as you showed with some of the data, that one batch had less interoccasion variability than the other. That shows the formulation has got, as a result of interaction with the physiological interoccasion variability, will demonstrate different interoccasion variability in the features of the PK. And that is the thing that I think we have to discuss more and make sure everybody appreciates the distinction.
Yeah.
Yeah, I think perhaps some kind of a new nomenclature to differentiate and clarify those and what specifically we are talking, it would help perhaps. Okay. Any other question, any comments before we close the session?
There was a question from Neil Parrott. Neil, did you want to read that yourself? On your...
Okay, thanks a lot. Yeah, I was just wondering if Ioannis had any thoughts on running VBE in pediatric population, because currently, we're asked to run VE studies for pediatric formulations in adults, and that seems a bit, less than ideal. So would it be feasible now or in the near future to run this for a pediatric population?
I mean, I suppose so, yes. Of course, there are some additional challenges, I would say, translating, typically scaling down the adult PK model to pediatrics, and of course incorporating any differences in terms of GI tract maturation or absorption related differences in the model, and how that would translate. So I mean, it's in principle I would say that it's feasible, but you have the additional steps that I guess they will bring additional complications. If you're assuming that you have some kind of, I don't know, interoccasion variability, or in adults, how this really translates in pediatrics, I would say that it's not straightforward. But yeah, that would be my thinking.
Thank you. Thank you. There's a reason I asked the question, but currently, I think what we do in running bioequivalence in adults and then give the dosage form to children is also less than ideal.
But I mean, even in adults, I think it's quite common, I would say, that we often neglect the interrogation variability, right? So, but as you're saying, it's even more complicated when you're going to go to pediatrics.
Okay, thank you very much. I think it was very interesting, as you can see, everybody, and interesting and I would say important topic. As Neil just mentioned, it is not only in the adult, but you can go to the pediatric, and then by extension you can go to a special population, so and patient population as well. So all of these they can follow up the discussions. I'm sure we will hear more about the virtual bioequivalence, given various parties, both in industry, in academia, as well as the regulators. They are interested in this topic, and the new guidances are coming out and addressing these various aspects. Again, thank you very much for Ioannis for the presentation. For everybody, before we close the session, we have another webinar is booked for 28th of April. Please add it to your calendar. We are waiting only just for the confirmation of the title. It's coming out. They are waiting to get the permission to announce it, and then we will have it. If anybody interested to present. Obviously, this one is open to any of you. If you are interested to present a topic, please get in touch with Oli. Oliver Hatley, he's organizing this. All the burden is on his shoulder. So if you are interested, please get in touch with him, and he will help you to arrange that one. Thank you very much, everybody, and thanks for the presentation, Ioannis, and have a good day.
Thanks a lot.
Thank you.